Okay, so here's what we're going to be up to today, guys. We're going to be doing a lab to investigate the stuff we've been talking about the last couple of days, which is how plants transport water. All right, so you're going to be using the equipment you see up here. Okay, so you're going to have a large graduated cylinder, a small graduated cylinder, okay, and some food coloring, and a piece of this plant. So we're going to hack up this plant. Don't worry, don't feel bad. It gets hacked up every single year. Okay, been using the same plant now for about 5 or 6 years. It grows real fast. Yeah. Okay. Just like hair, right? You get a bad haircut, it grows back. It's no problem. So what we're going to be doing here, guys, is essentially dyeing the water. You probably did this like when you're in grade 5 with a piece of celery. Okay, it's kind of a similar thing, except this time we're going to use a real plant. Well, not that celery is not a real plant, but okay, you get the idea. All right. So the problem we're investigating here is, what are the forces that move water through plants? Okay, there's some background there, about scientists marveling at the ability, blah blah blah. Okay, but the general thing here is that what are the forces that move water through plants? Well, we talked about those yesterday. Okay, there's no pumping force there. Okay, there's just the force of osmotic pressure, that osmotic pulling force from above. Right? We're going to investigate whether that's actually the case. The way we're going to do that is we're going to eliminate all other sources of possible forces from this plant. The piece of the plant you're going to cut off won't have any roots on it. So we eliminate the roots as a source of pumping force or whatever to move the water. The only thing left then to move the water is things within the leaf. All right, essentially our osmotic pulling force. So that's the purpose of just cutting off a piece of this plant. Okay, now the way you cut that plant off and the way you put it in the water, it's really important that you do it right. If you don't, you risk getting a lot of air into the xylem, and then the plant kind of, it locks and it can't get stuff through there because the polar nature of water has been disrupted by the air bubbles you've allowed in. All right, so what we want to do is try and eliminate that as much as possible. I'll tell you how we're going to do that. All right, so manipulated, responding, controlled variables. A little bit tough on here. Are we changing the plant in any way from its natural state? Yes, we are. Okay? We're manipulating the plant here, right? Specifically, we are manipulating how it's able to move stuff, or we're eliminating at least parts of it we think could be suspect in terms of pumping water as opposed to just using the force we think. All right, so we're eliminating parts of it. Okay, we're changing essentially the structure of the plant a little bit in order to isolate the force we want. Right, responding variable. Here's what we're going to be watching for. We're going to set this up today. We're going to record how much water is in the small graduated cylinder, and the plant will be sitting in there. Right, and then on Monday when we come back, hopefully there'll be less water in here. Right, so what are we watching for? Yeah, we're watching for the change in the amount of water in the graduated cylinder, but we're also going to be looking at any changes in appearance of the plant, because if it's drinking up dyed water, what should we see in the leaves later? Yeah, we should see color moving into the leaves in certain places, into the veins, and believe it or not, on the bottom of the leaves by Monday, you'll actually be able to see where all the stomates are. Okay, because where does the water move to? To the stomates. That's where it evaporates from. Right? So that's what the kind of thing that those are the things we'll be looking for, right, as responding variables there. Controlled variables, well, okay, we're going to do our best to have all the plants in the same place. They're all going to sit on the windowsill. All right, well, actually in the biolab. They'll all sit in the windowsill, so they'll all get the same kind of environmental conditions. You need to list off anything you think will be the same for all of the plants throughout the weekend. All right, remembering that this is a temperature-controlled building. Okay. All right, so the hypothesis here needs to talk about how plants move water. What part of the hypothesis should talk about how plants move water? Okay. Probably the F part. Okay. And part is what we're doing, describe our experiment. Then part describes your expected findings. So you tell me what you think you're going to see on Monday as the then part of your hypothesis. Everybody got me? Okay. Okay, materials, you can write C sheet, same as always. Okay, you're going to set up your experimental apparatus like I'm going to show you here now. Had some pictures of that from last time. This is about as big a piece of the plant as you need, so let's say 3 leaves at the most. All right, so you're going to have to maybe go back a little ways onto the plant, all right, to get, 3 leaves, right? Yeah, probably a piece like that with big leaves like that, you probably only need 2, right? So you'd have to kind of go back to here on the plant. But here's the key. I'm going to fill the front sink up with water, and I'm going to stick this plant in it. All right? You need to make your cut under water. All right? So submerge the plant, the scissors, cut it under water, and then stick your thumb over the cut before you bring it out of the water. That way, we're limiting the amount of air that's allowed to enter the xylem. All right? Now, a little bit of air is going to get in there when you transfer it to your graduated cylinder, right? But not as much as if you wander around the classroom with the end open for a couple of minutes on your way there. Everybody follow? Okay? So you do have to make sure of that. That's the most important thing in terms of making this lab work properly, is to not allow any air in there. So, okay, we get a plant kind of like that. All right? Here's the cutting procedure. I think we took pictures of everything last semester. All right. So underwater, make sure you're underwater when you cut it. Okay. And then you put a couple of drops of dye. Now here's the other thing. We need to be able to know exactly the volume inside of this graduated cylinder. But the problem with these graduated cylinders is they only have markings about 3/4 of the way up. And we need to fill them pretty close to the top. Otherwise, there's a chance that, you know, the stem of our plant might be out of the water by Monday. So we need to make sure that they're filled all the way to the top so that our plant, which may only go down about this far, okay, will still be in the water by Monday. Everybody follow? So to measure that, that's why you need 2 graduated cylinders. Okay? You fill the big one up with water. Tap water will do. Okay? And put, let's say, 3 drops, 4 at the most, of food coloring into that. Swirl it around or use a stirring stick if you want, okay, until you've got it nice and dyed. Now, this is a 25 mil graduated cylinder. I would say to fill it near the top, be on the safe side, make 60. You won't use it all, okay, but make 60 mils, mix it all up, and then pour it in here until it's full. Okay, now you need to make exactly 60 so that when this thing is full, you can look at how much is left in here and know how much is in this one. All right? That's the only way you'll know your initial volume, is to have it on the graduated cylinder that's got enough markings on it for you to know. Everybody with me? Then on Monday, we'll reverse the process. You'll take your plant out, you'll make your observations, and you'll dump this back in and record how much is in there now. All right? And it will be less. Not very much less, but it'll be less. All right? Does that make sense to everybody? Okay, so what you need to record today when we're in the lab is what your plant looks like. Maybe take a picture of it with your phone. Okay, and how much water it has in it to start. Okay, that's essentially all you'll record. Those are your initial observations. Once we've got that all set up and the plants are in the windowsill, then we'll come back here and work on the lab report. One last thing that I've forgotten to mention. This is open at the top. Does that prove troublesome? Yes. We need to seal that off. All right? Otherwise, we've got all this area where water will evaporate from, and that's not a very good controlled experiment. So, I will be giving you guys the finger from a rubber glove to put over the top of this graduated cylinder. All right? Creates a nice tight seal over the top. You just poke a hole in it with your pencil and push the plant through the hole that you made. All right? Don't make a big hole because, again, we want to make as airtight, it's not going to be airtight, but as airtight as possible of a seal around the top. Everybody got me? Okay, so what you see here is them just adding the blue drops in, okay? And then this is kind of the final product. And you can see in this picture right here, this is the finger, okay, from the rubber glove, okay, that's just wrapped and kind of down over the length, about half the length of the cylinder. Okay? Everybody got me there? It's not too tough, all right? But you got to follow the instructions pretty carefully. Okay, so yeah, and that was just me explaining all of that stuff. Okay, we'll talk about the analysis interpretation later. All right, questions on any of that? What are you recording today? How the plant looks and the amount of water that's in it. All right, let's head across then and get that done. All right, so we've done all the procedure part now. Now we just have to wait, okay, you know, over the weekend to let the plant carry out photosynthesis and start moving some water. Okay, probably by Monday those plants will start to look kind of sick. right? I mean, obviously they don't transport water quite as well without their roots because they don't absorb it quite as well, right? But they'll do the job. So some things to think about here in terms of analysis and interpretation. Okay, we want to be able to calculate the amount of water taken up by the plant. Okay, obviously that'll be a fairly easy calculation on Monday. Just compare your initial and final values. Okay. What evidence do you have that water followed the path discussed in class? So the path we discussed in class, okay, was, you know, through the xylem and all that kind of stuff. So review that and then explain to me what evidence you have of that. You should have pretty good evidence of it on Monday when you look at your plant. Okay, but again, be thorough when you're describing that. Okay. And then thirdly, predict the effect of adding salt to the water and describe how you could test this prediction. All right, now, so you need to go into detail and explain to me, if I added salt to the water, this is what would happen, and explain that based on how you know a plant pulls water up the plant. Okay. Then your conclusion, restate and explain your explain why you accept or reject your hypothesis. Identify your sources of error. Okay, you don't have to do the class discussion questions. We'll talk about those when I hand back the lab. All right, are there any questions there? So what you have time for right now, guys, is to get all your variables, hypothesis, done. Okay, now you don't have time for that. So there you go. That's what you got. We'll discuss that next week after we actually have the data.